everyone, welcome back to lecture 4 of part 2. And apologies if I seem a little bit sluggish, second day of Ramadan, always a bit tough. So to those who are fasting, all the best to you and have a great Ramadan. We basically covered immune response to infections, immunological memory, and vaccination. And then in the last lecture, we talked about immunodeficiency. So we talked about what the immune system is supposed to do, how it does its job, what happens when it's deficient. And now we're going to talk about a slightly different take, which is immunological tolerance. So what is actually tolerance? We'll find out. At the end of this lecture, I hope you'll be able to explain the concept of immunological tolerance and describe the central and peripheral tolerance mechanisms and the role of regulatory T cells. Now, I always like to use this analogy for immunology. You know, our immune system is basically our defenses. And it's very easy when you want to defend your territory. You just look for what is different, what is the other, and then you attack that. And that's how you think you're protecting the body. And in order to know who is the other and who is actually on your side, you have to recognize who is on your side, right? And then if you look at this picture, you've got one side, right now they're separated, so you would assume the one that's on this side is on one side and on the other side is another group. But once they start merging and they start battling, it's very confusing. And so things like having a similar uniform, having certain badges, maybe having a dragon on your side, that kind of helps you identify who is on your team and who is not. And this is basically the gist of how our immune system also is able to function so that they don't attack the cells that are on their side. Very early on, we know that any self-reactive T cells or self-reactive B cells won't even survive, will not even enter the circulation. So very early on, we know they're removed, and this is supposed to give us both central tolerance and peripheral tolerance. But what is tolerance? So I like this word tolerance. It means you don't respond aggressively. You, in a way, you're more patient, you're bersabar sikit, and we talk about tolerance a lot as well in human societies, right? Even though we're different in a lot of ways, it doesn't mean everything somebody does that's a bit different from us makes us get angry and makes us respond aggressively. We try to understand and we try to tolerate, and even better, we try to integrate where we can, yeah? And this is the idea of living harmoniously. And in the same way, even in our body, the most healthy type of response is the one that also has some tolerance, whereby it will not attack its own cells that carries its own antigens, but it will be able to tolerate some foreign antigens as well that technically don't have any potential for harm. So the immunological tolerance, by definition, refers to the layers of protection by the immune system to prevent immune attack on its own cells and tissues. And previously, as you saw in the slide before, it was thought that it's just achieved by eliminating cells that can react against self-antigens. So what you have is just a state of unresponsiveness to self. So they're basically not even there. Cells that can recognize their own cells as potential enemies are not even in there, in circulation. So that's the previous idea. But new studies show that it's actually not as simple as that. There are some cells that will escape that initial self-reactive deletion and enter the circulation. So instead of ignoring self-proteins, the immune system now tries to protect self-proteins. And this is what led us to the discovery of regulatory T cells. So regulatory T cells recognize self-proteins and protects them. So it's a pretty cool idea. So if we revise that figure, you've got now the central tolerance is when you get rid very early on of any self-reactive T cells or self-reactive B cells. But for the peripheral tolerance, you actually select self-recognizing cells that can protect instead. So we'll go into a bit more detail on how this actually works. But it really relies on this subset of T cells called the T regulatory cells. And what it does when it recognizes self-antigens is that it inhibits the immune response. So very clever. But first, the central tolerance, which is what is the prevailing idea, and it's still true, is that basically a lot of the tolerance is developed in the early stages of lymphocyte development, even in the beginning when we talk about clonal deletion, right? So that happens because any of the clones that's carrying a particular receptor that can recognize the self-antigen will be deleted. So it's a process of negative selection and receptor editing to eliminate any autoreactive T cells. Sorry, not any, but many, not all of them, but many of them will actually be eliminated at a central level, either in the thymus for the T cells or in the bone marrow for the B cells. So the two mechanisms that helps us achieve central tolerance is, as I mentioned, the negative selection. So if they recognize a self-antigen, then that clone gets deleted, and especially if it recognizes self-antigens with high affinity. But there's also another process where, let's say the B cells recognizes a self-antigen, it can also go antigen-specific receptor editing, so that it kind of changes the receptor that binds the antigen, and that allows the B cell now to pass the threshold of self-recognition, and can still allow that cell to survive. So kasiyan the cell. It's not its fault that it's carrying a self-identifying receptor. So we change that receptor a little bit, and now it can be allowed to continue to develop and enter the circulation. Sorry, little pause there. Right, so this is the schematic that shows the central tolerance that occurs. So we see here a lot of the cells that recognize self-antigens in the development will be deleted. And then the clones that do not recognize the self-antigens will enter the primary lymphoid organs. And then you also have a subset that are able to suppress self-antigen recognition. So that's our T cells, our regulatory T cells. And then we'll talk a bit more about the peripheral tolerance. So the peripheral tolerance refers to the regulation of lymphocytes after they've entered circulation. So in the event that you have self-reactive lymphocytes that were not deleted for some reason, you have peripheral tolerance to kind of help top up. So why do we need it? Because, as I just mentioned, not all self-antigens are expressed in the central lymphoid organs where negative selection occurs. So you will have some self-reactive lymphocytes that enter the circulation. And then there's actually a threshold requirement for affinity to self-antigen. So it's not just recognizing it. It has to recognize the self-antigen with a certain level of affinity, and that will trigger the clonal deletion. So you might still have some weakly reactive clones that didn't get that recombination that we mentioned earlier, still able to survive and enter the circulation and possibly cause problems down the route. So there are a few known mechanisms for peripheral tolerance, but the most important one that we're learning more about now involves the T regulatory cells, which is a subset of CD4 and CD8 cells. So the right. So this schematic here shows what happens to the T cells that, I mean, we've already seen a version of this before, and we know that the ones that have high affinity for self-antigens will be deleted, so they'll undergo apoptosis and commit suicide. The ones that have low affinity for self-antigens, though, they might actually still get through. So they are these weakly autoreactive T cells, and that's why we need peripheral tolerance as well. And that's achieved when we have these other T cells that have an intermediate affinity for self-antigen, and then they actually have an upregulation of FOXP3, which becomes a key thing with these Treg cells, which now are able to recognize and suppress the T cells that are able to react to self-antigens. So the peripheral tolerance by Tregs. So the Tregs, previously they were called suppressor T cells, because they technically do induce immunosuppression, and they downregulate induction and proliferation of effector T cells. But now they're better called regulatory. So they just regulate the behavior of these other immune cells. They're a unique subset of CD4 T cells mainly, but there are some that are CD8s as well. And they express high levels of the CD25, the high levels of CD25, which recognize the IL-2R. So what people noticed was that in mice that are deficient in CD4 Tregs become very susceptible to autoimmune encephalomyelitis, so a type of autoimmune disease. And the activity of regulatory Tregs are usually in the secondary lymphoid tissues and sites of inflammation. So the Tregs will recognize the specific self-antigens, and it also recognizes some foreign antigens through the T-cell receptor interactions. But when they recognize these self-antigens, or even some foreign antigens, they downregulate the immune process. They don't ramp up the immune process. So this is what is called that suppression. So it suppresses the immune response. And this also is important when you think about allergies, because allergies is when our immune system overreacts. Guys, hold on.
There's a bug.
Shh. Where are we? Right. So yes, so as we know, even with allergies, it's basically an overreaction. So the Tregs play a big role in this as well, where they try to recognize some of these harmless non-self antigens to reduce the immune response when our body encounters them, especially if they're coming from things like the commensal microbes that make up our gut flora. So this is, I think this is why they changed the name to regulatory rather than suppressor. Why? Because they do so much work. Like they're able to
not going somewhere far.
Ma. Sorry about that. So they do a lot of regulation for different types of cells, like the effector T cells, even dendritic cells, and they recognize different receptors, and they will also upregulate different molecules in response to these. A key part of peripheral tolerance also is the CTLA4. So the CTLA4 is an interesting molecule which is able to induce energy. So in order for T cells, to become activated, the T cell receptor has to bind the antigens presented by the self MHC, and then they'll undergo post-stimulatory engagement. So when the CD4 T cell clones are actually only stimulated to the DCR alone without co-stimulation, they become energic. And this is when to avoid this, it's usually because you have like CD28 on the T cell and CD86 on the APC, the antigen presenting cell, which provides the co-stimulatory signal required for activation. Now with the CTLA4, that's the molecule that actually inhibits rather than stimulates the T cell activation when it binds to the CD80 or 86. So the CTLA4 is induced when the T cells are activated, which then actually gives a mechanism to stop the T cell to get activated and become effector T cells. And what you notice is that mice that don't have the CTLA4 have a lot of autoimmune diseases. So this is basically a schematic which I hope is more clear to understand. So you have the APC presenting the antigen. Here, let's say this is the self-antigen, and the T cell recognizes it. But then because of the CTLA4 that binds to the CD8086, it actually induces energy in the APC. So, or it changes type of cytokine profile that the APC produces, which makes it less immune reactive and more immune suppressed. So there are also regulatory CD8 T cells, and they also play a big role in inhibiting responses to self-antigens. But unlike the CD4 Treg cells, it's a very small population of natural Tregs from CD8 T cells. So this is actually a very useful schematic for you to look through in greater detail. And basically, these are the different peripheral tolerance mechanisms that a T cell can intrinsically go through. So you have the first one here, A, it's just ignorance. So the T cell here has a receptor that can hypothetically recognize a self-antigen, but it never actually meets this self-antigen. So it survives in a state of ignorance, never meeting what it was meant to bind. Then you've got the case where the T cell encounters the self-antigen, but via the CTLA4 or the PD-1, instead of activating the T cell, it actually induces the T cell energy. And then you've got T cells that undergo phenotypic skewing. So when it interacts with a self-protein, it activates, but it fails to induce autoimmune tissue damage, and this is because it has a change. It induces a change cytokine or chemokine profile. And then finally, the last one, it could be because the self-reactive T cells, when it meets a self-protein, it is induced to a cell death involving the FAS protein. Right, so peripheral tolerance regarding, so just now we talked a lot about the T cells, but basically what they found through experiments is that when mature B cells encounter most soluble antigens in the absence of T cell help, they become anergic. So there are some that are T cell independent, but most of them do require T cell help. So in a way, if you have a T cell tolerance to these self-antigens, it also makes the B cells tolerate the same antigens. Alright, we're nearing the end now, so let's talk a bit about what are tolerogens. So tolerogens are antigens that induce tolerance rather than immunogens, which induce immunity. So the tolerogens induce specific immune non-responsiveness. And what's interesting is that this can be the same antigen. An antigen can be either or both an immunogen and a tolerogen, and it really depends on how it is presented to the immune system. So some antigens, for example, when they're introduced orally, they will produce tolerance, but if you actually inject it, it becomes immunogenic. So the route of administration is quite important in determining tolerance or immunogenicity. Yes. In other instances, if you mucosally administer the antigens, it gives protective immunity. For example, the oral polio vaccine. So that one's actually more protective compared to the injected, but that one's also an inactivated one. So, and tolerogens are also antigen specific. So just because you inactivate the immune response, it does not result in a general immune suppression, but just that when it encounters that particular tolerogenic antigen, the immune response is not stimulated. So here's a comparison of a normal immune response versus tolerance. So here's your lymphocyte and the receptors. It encounters an immunogenic antigen, bam, it gets activated, and then it proliferates, differentiates, become effector cells. But if it actually encounters a tolerogenic antigen or a tolerogen, that induces anergy, which means it doesn't respond. So it will either cause deletion or cell death, or just no response, really. Right. So a bit about adult versus fetal antigen response. What was found is that most adults, when they encounter foreign antigen, they will develop an immune response to eradicate, because their immune system is a lot more developed. In the fetus, because it's less developed, sometimes being exposed to an antigen frequently results in tolerance. So this is where you have sort of what the baby is exposed to in the womb by virtue of what the mom's eating or the environment that the mom is in. It will make the baby more likely to tolerate those things when they're born. This bias towards tolerance is also seen in transplants. So you have a higher success rate in young children compared to older children because the antigens tend to not stimulate an immune response, so it doesn't result in rejection of the transplant. It results in tolerance to the transplant. So I showed in the schematic earlier, one of the outcomes of self-reactive T cell that enters circulation is that it might encounter its self-antigen and then it undergoes apoptosis, so programmed cell death. So this is an important part in both central and peripheral tolerance. And activated T cells express increased levels of FAS and FASL. So in both B and T cells, when they're engaged, they will cause activation-induced cell death. And the mice, in experiments that carry inactivating mutations, they're not able to engage this pathway and they develop autoimmune diseases early in life. Another important thing about tolerance is the role of gut microbiome. So usually what they've found is in mice, if there's actually germ-free mice, they fail to induce an effective immune response and fail to provide protection. So the full maturation of the host immune system is dependent on the natural harboring of host-specific microbiota. So a bit of a mouthful, but it means whatever that you're carrying in the gut is going to influence the development of your immune system. And in the beginning, your gut microbiota is actually not very stable. Up to 3 years as a child, any disruption to your gut microbiota, for example, if you take antibiotics as a baby, that can significantly alter your gut microbiota in the long term when you enter adulthood, and so that will impact your immune system as well. Okay, last bit, the role of maternal antibodies. So we know that babies get a lot of maternal antibodies through breast milk, so that's a lot of your IgAs, the secreted IgAs. So this helps establish the microbial composition, and it also dampens excessive T cell and B cell responses to gut microbes. So it helps, it helps, not yet, I'm not done yet. I'm still recording, but yeah, I don't know. So it's inducing tolerance and preventing intestinal inflammation towards the gut microbiota when you actually have breastfed babies. So breast milk is a very good source of a lot of goodness. So part of the gut tolerance is also the role of the Tregs and the Th17s. They're very important to establish the tolerance to food antigens. So as I mentioned, the Tregs recognizes our self-antigens and some non-self antigens, but those that belong to what we call the harmless antigens, like food antigens. So in certain mice, the Th17 cells are the most abundant effector CD4 T cells, and they provide tolerance to commensal bacteria. So they promote survival of antigen-specific IgA B cells, and the depletion of the Treg cells with anti-CD25 results in loss of intestinal IgA B cells. And that will, the failure of Treg induction will increase susceptibility to intestinal inflammation. So you can have a look at this diagram just to show sort of how the different T cells are involved in the microbiota immune regulation. So you've got the Tregs here with their FOXP3 and how they deal with the antigens from the microbiota, the Th17s and the Th1s. So basically the Tregs will induce the tolerance compared to the other two. Another way to actually avoid self-reactive responses is sequestration or compartmentation of antigens. For example, in your anterior chamber and the lens of the eye, your circulation will not actually access any antigens there. So it's a sequester site. So you can actually sequester a lot of antigens and isolate them from interacting from the immune cells. So it will allow those T cells that would have been self-reactive survive in a state of ignorance. So they just never encounter their self-antigens. Some other things that promote tolerance include high doses of the antigen, long-term persistence of the antigen, the host.
Now say something
So again, if there's a lot of them, you're like, okay, I guess I will just tolerate it. Or, okay, they've been here for a while, I'm going to tolerate them now. Specific routes, depending on the antigen, whether IV or oral introduction. Absence of adjuvants. So adjuvants are some things that actually do stimulate the inflammation. And if they are not present, then you don't really get as much a robust immune response. Or as we mentioned earlier, if the co-stimulation doesn't work very well, or if the presentation of the antigen is by immature or unactivated APCs. Alright, so that's the end of it. Sorry about this lecture, very many disruptions. This is work from home for you. Think about whether or not you want to have children, if you want to have a career. So let's just recap very quickly. Mechanisms to prevent self-reactivity is tolerance, to operate at several levels. And the first level is the central tolerance, which serves to delete self-reactive T or B lymphocytes. Peripheral tolerance inactivates or regulates self-reactive lymphocytes that survive the initial screening process. And largely that's regulated by T cells and involves the CTL4 and the fast cell. And the Tregs that are characterized by their presence of the FOXP3. Okay, thank you very much. Have a good week.